Imagine you find a magical copier that can duplicate any book you feed it in no time. But what you quickly find is every time you copy this book, parts of the story get deleted. More specifically, every time this book is copied, a chapter from the beginning and the end of the book is lost until there's nothing left to copy. In our cells, the enzyme DNA polymerase is like a DNA copier, and it suffers with the same problem as our magical printer. DNA polymerase cannot copy the ends of linear DNA strands. Thus, with every division, our story gets a little shorter. Welcome to the last generation to age in our series on the hallmarks of aging. In this episode, we'll be breaking down one of the most famous of hallmarks, telomere attrition. We've spoken about how DNA is packaged. It is supercoiled around proteins to form linear chromosomes. As mentioned before, DNA polymerase cannot copy the ends of linear DNA. This is called the end replication problem. Thus, with every division, our chromosome gets a little bit shorter at both ends. This is where telomeres come in. Telomeres are long stretches of non-coding DNA wrapped around proteins that protect the ends of DNA. In our copier metaphor, telomeres would be like adding disposable blank pages to our books. The books still get shorter, but we don't lose any of the important story. Another problem with linear chromosomes is that the ends look a lot like double-stranded breaks, which are a form of DNA damage we mentioned in our episode on genomic instability. The DNA repair mechanism can detect these ends and join them to other chromosomes, which we do not want. To stop this, the ends of telomeric DNA are tucked in and hidden by a group of proteins called the shortening complex. The bad news is, telomeres still shorten, and while some cells, like stem cells and cancer cells, rebuild their telomeres with enzymes, in most cells, telomeres shorten until they become too short and the cell stops dividing. This imposes a limit on the number of times a cell can divide, a limit discovered in 1961 by Leonard Hayflick, aptly named the Hayflick limit. Not surprisingly, just like our previous hallmarks, genomic instability and epigenetic alteration, this leads to cellular senescence, which we'll go into in full detail in a later episode. We mentioned that some cells, namely stem cells and cancer cells, overcome the Hayflick limit by building their telomeres back with an enzyme called Telomeres, which we'll cover in our series on treatments. Unfortunately, simply giving telomeres to normal cells that have passed their limit potentially resurrects highly mutated and cancerous cells, presenting a major hurdle. Thank you.